people after the seminar. So never get the bus. Yeah, because it's still a boy. But just so empty the house, so 2-3 more lives. No? Yes. Okay. Sorry, I should have to stop my little chat with Indrani. Thanks for coming to our seminar. It's great to see so many people. I think this is probably the biggest group that I have personally seen this semester. I hope this continues like this. I have not much to say. I have some random good news to spread, perhaps. First, the more social thing is you have maybe all seen that and Rani wouldn't know that, but we've had a big hail damage from like about 4 or 5 years back. Every single roof on the ANU campus was affected, and then of course it all had to be restored to its original beauty. And we were lucky enough to have a heritage listed copper roof on Jaeger 6, I think it is. And this has now been restored to its original beauty. So even if you are tempted to use a little bit of copper for capsules or for electricity fixtures, do not touch that roof. Then the newsletter will have a couple of really nice pictures, and there was a drone movie made the other day. So at least we can marvel while it's still shiny. And we have already plans to make it green, right, Jochen? But we cannot talk about this, not in an open forum. Otherwise, I have what I can say is some good news from our ECR cohort. We have Kellyanne Lawler, who's probably not here today, who's a PhD student, and she has just won the Leanne Armand Travel Award. The Travel Award is, of course, named after our beloved colleague Leanne, who passed away too soon and quite brutally. But with at least to honor her memory, there is this Travel Award, and Kellyanne is the recipient this year. And then we also have Claire Young. Claire is also not here? Okay, right. Claire's already on her way to Germany because she was selected by the Australian Academy of Science as one of the 10 ECRs from Australia to attend the 73rd Lindau Nobel Laureate meeting in Germany. So Lindau is a beautiful place and is probably in full bloom. And this is certainly a fantastic honor as well for Claire and for us for her to actually talk to the Nobel laureates there and to fellow ECRs. So now you know more than anybody else who does not come to the seminars. Do not tell the others. Let them stay ignorant. And now we come to Indrani's wonderful seminar, and Michael is going to introduce her.
Yeah, so thank you all for coming today. I'm very happy to introduce Indrani Mukherjee, who came here all the way from sunny and warm Sydney. Before I begin and forget, there's a dinner sign-up sheet on the door outside with a pen. So if you would like to join the dinner, it's at 6 p.m. In Lemongrass. Please sign it so I know to call them and book a table for that amount of people. Very important. Other than that, she is also speaking tomorrow in the Petrology Group seminar at 12:30 on the exciting topic of pyrite. So for those who are not on the list and are interested, you can talk to me and I'll give you the information for that. Now, I have a very long page here with an introduction. So I will start by saying that Indrani wanted me to say that she is the ultimate pessimist, and also some other things, which never mind now. She earned her bachelor's and master's in geology in Delhi, in India. Then she did her PhD in Tazi. That was with Ross Large, right? Yes, Ross Large. Then she had a short stint in Toronto, and then she got a job as a lecturer at UNSW. Her interests are the transition of simple cells into complex ones billions of years ago and millions of species today. I probably messed it up, but I'm pretty sure that we will learn about this. I can talk some more, but I won't. So please, Indrani, please tell us interesting things.
Is that all right? Good afternoon, everyone. Thank you very much for inviting me, and thank you, Michael, for giving me the opportunity to come by and share my research. The topic that I've chosen is very, very close to my heart, and hopefully you'll see why. Just a very quick correction on the introduction. Sydney right now is not sunny and warm at all. So, but now that I'm here in Canberra, yes, it does. Maybe it is sunny and warm back there. But no, it's a real pleasure to be here. I've been here at ANU. This is not my first stint, and I think I specifically mentioned to Jochen last week that I was going to share this story about my first stint at ANU as a PhD student at the shrimp lab. There was a PhD student there, and he said, your topic is very close to Jochen Brox's. Do you want to give a seminar? And I was like, oh my God, Jochen Brox, he's here? Yes, sure. And he's like, yeah, if we advertise your talk, I'm sure he'll come. And so things happened, and the David Green room, I gave my seminar. There was one person who walked in slightly after the talk started. Anyway, the talk ended, and there was one guy who asked a lot of questions, a lot of good questions. The seminar finished, everybody left. The guy who asked the question was still sat in the room, and I said to the seminar organizer, hey, by the way, where was the Brox guy again? Like, I never, and there was some guy on the computer just said, oh, sorry, I forgot to introduce myself. My name is Jochen Brox. And I remember I just felt the earth underneath me, just like someone just drown me or do something. And poor Aditya, who was the PhD seminar, just was like, that's him. So, yep, no, we still talk to each other, and yep, hopefully he doesn't remember it, but I did remind him last week. So yeah, I thought I'd share this story with you, that this was roughly 10 years ago. And so here I am back talking about that same old topic, which is never going to get boring for me, if you ask me. So we're specifically referring to the boring billion period, which is commonly known as from the 2000 million years old to 1000 million years old, or 1.8 to 0.8 billion years, whichever way you look at it. So is it boring, balanced, or brilliant? Hopefully you'll get a balanced view from me, but I seriously doubt it. You can make your own opinions after my talk. It is customary these days to do acknowledgement of country, and I'm an immigrant in this country, so I am very mindful of the fact that I've been given an opportunity to pursue my scientific career in this country, and I really am grateful to our First Nations people of Australia to give me that opportunity. Other than that acknowledgement, I'm probably not doing much, but some of my colleagues are, so I thought I'd share this information with you. My colleague at UNE, she produced a movie where she brings along geologists and First Nations people, and hopefully I should follow her path and do something more important than just saying that I acknowledge the First Nations people. So so it really bothers me that I say that, but I'm actually not doing as much as some of the other people are. But it's a fantastic movie if you'd like to watch it later on. So, like I said before, the boring billion saga for me started when I was doing my PhD, and I thought it was going to be a pretty simple thing. One of the first things my PhD advisor did was take a sheet of paper and 1, 2, 3, I don't know, ANU somewhere down here, I guess, to go and do some shrimp analysis. And it looked pretty simple at the time. And I thought, surely it's a series of steps and I'll just do it and it should be fine. And something he said a lot was, take it easy, but I'm sure he never meant it. So that's something, if there are any PhD students here in the room, and if your supervisor is saying, take it easy, they probably don't mean it. So boring billion, like I said, the geological time scale I don't know if it's 2 to 1 billion years or 1.8 to 0. But it's roughly that time. And specifically the Mesoproterozoic, it gets all kinds of negative connotations. And I can see it. Why? Because the Archean, you can talk about the origins of life forms, lots of different mineral deposits, and of course, the birth of the planet itself, right? So there's lots to talk about in the Archean. The Phanerozoic, my God, you've got dinosaurs. I mean, honestly, that's what fascinates every person on Earth. All you need is a big fossil of dinosaurs and people are going to go crazy. So fair enough. Then, of course, you've got the great oxidation event on either side of the boring billion. And then, of course, the new Proterozoic is known for our ancestors, the Metazoans. So the boring billion gets a bad rep because of that, because of this comparison, I think. And so, like Lyons et al. Would pointed at in his 2021 paper, that it's bracketed by some of the most striking transitions in Earth's history. So that's why the boring billion time span, in my opinion, is getting this really bad reputation because of what's going on either side. And of course, let's address the elephant in the room straight away. It's the macroscopic life forms. That's, after the boring billion, and everyone is an explosion of life form, Cambrian explosion, this, that, and humans, of course. So yes, everything interesting and everything big occurred after. So something must have delayed evolution, I think, was the concept when it was introduced by late Professor Martin Brazier in his 1998 paper, where he coined the term boring billion, not so much in the paper, but in an interview with a journalist. But there's a story behind that as well. But I think what he looked at was carbon isotope records through time, and he observed this prolonged stability or sort of non-fluctuating delta carbon 13 trend across the boring billion. And he assumed, or he thought, that perhaps implies some sort of stability in our oceans. That must have sort of a dull period, which perhaps delayed evolution of life. And once that period was over, you get all the explosion of life forms, etc. And soon others follow suit. There are lots of papers, by the way. I'm just going to just comment on a few of them, the ones that were quite important for me when I was doing my PhD. But yeah, they gained a lot of traction, this term boring billion, and everybody jumped on board with whatever tools and techniques they were working on and saying, well, what does my proxy tell me? What happened back in the day? So Scott et al. 2008 looked at molybdenum concentrations in marine black shales through time, and again observed that, yeah, not much happening, low molybdenum concentrations implying low oxygen in the atmosphere. Sure, that would have fueled the delay in the evolution. And there were lots of different papers, and including my own supervisor, who said life on Earth was nothing but slime for a boring billion years, and he was my advisor. So I was like, well, I don't know, why am I even working on this field? Is it known that it's boring? So we know that. And he was like, well, no, you just see, let's use our technique. And I'll go into that a lot. But again, I think a lot of the misconception was, well, then nothing happened in terms of evolution of life. So biological evolution was at some sort of standstill during this period. And it's only after the boring billion. I'm going to try and get the pointer. So it's only after this period was over there, you start seeing action going on in Earth's history. So I started with that sort of background info. And so for today's talk, I just wanted to go through some of these individual little subsections, because if we're looking at biological evolution through time, I think that geochemistry alone can't answer it. So I can work on my technique and find some answers, but ultimately it's tied to the broader scheme of things. And one of the main things would be plate tectonics. Well, where were we geologically speaking? Like, what were the geodynamic conditions? Of course, atmospheric and ocean chemistry will have been really important too. And then what actually happened in terms of, what do we know? What happened in terms of biological evolution? How much do we know? So we'll go through that one by one. And so in terms of plate tectonics, if you do a little bit of reading or search in literature, you'll find that really in the Proterozoic, we genuinely formed Earth's first supercontinent. Prior to that, you had kind of an amalgamation of Archean supercratons, but the Earth's first major supercontinent, Nuna or Colombia, whatever you like saying, it was actually formed in the Proterozoic, in Paleoproterozoic. So to say nothing happened is again a sort of a misconception. And not me, but lots of researchers that I've collaborated with over the years have looked into this and have come up with their own explanation as to what happened when Nuna formed. Did Nuna break up to form Rodinia like it would normally supercontinents do? Or was there lid tectonics? So there's all kind of controversies, but I think more and more evidence suggests that Earth's first supercontinent did form during the Paleoproterozoic. So we've got that happening. So bear that in mind, because that has enormous consequences of what's going to get eroded and be supplied in our oceans, and basically create the ocean's first repertoire of bioessential elements. So if we think about it, the plate tectonics side of things will have a huge impact on what we deliver into our oceans, and then nutrient concentrations, et cetera, et cetera. So we did have the continents for sure, and the starting materials for the boring billion oceans. Just recently, I read my abstract yesterday, and I said I will present the latest research. So I was like, I better look up in the meantime, should something have come up. And I came across this little commentary paper on the balanced billion. So now we've moved from boring to calling it the balanced billion. So there's a lot of work being done on what the deep Earth might have been like. I'm not a igneous petrologist at all. In fact, I was quite surprised when Michael was showing me his lab, which looks very different to our geochemist labs. But yeah, so people are looking into what the deep Earth might have been like and how that impacted the overall tectonic cycle. And so it comes across as, it's not like nothing happened, far from nothing happened. It's actually a very stable situation, which they are now calling it balanced. And I think that also has important consequences of how it relates to my own data. And of course, oxygen. That has been the central focus of most deep time geochemists proxies that work in my field, especially in terms of the boring billion system. Well, it's a lot of emphasis on what the oxygen concentration was in the atmosphere. Was there enough oxygen to sustain life? Maybe there wasn't. That's why it's the boring billion, et cetera, et cetera. And the thing is that most of these geochemical proxies, including the one that I use, which I will come to, underpins the fact that it's related somehow directly or indirectly to the concentration of oxygen in the atmosphere. Because the more the oxygen concentration in the atmosphere, the more the rates of chemical weathering, the more you weather the rocks and you combine that with erosion, you transfer a certain suite of elements into the ocean. Now, that depending on the suite of elements, whether or not it's nutrient element or a non-nutrient element. But a lot of emphasis on oxygen. But importantly, if you think about it, we don't actually know what other atmospheric constituents were doing. Just because you have high oxygen doesn't necessarily equate to biological evolution or that it'll have some biological impact, because right now in our atmosphere currently, oxygen is not the highest concentration. I think it's nitrogen, but we don't necessarily use nitrogen for our respiration. It goes straight in and out. So higher availability of something does not necessarily equate to its higher utilization, is something that I was trying to understand. Of course, in some instances it is, but in most instances it may not be true. So that's something that needs to be looked at. And the other interesting thing is, I started my PhD in 2014. Was it? Yes, 2014. And it's in 2021, you can see how the graph's changing. And that also gives you an idea that as we progress, you're getting more data, you're looking at things differently, and the story changes every now and then as well. So now we know that there were these little peaks of or little bumps in oxygen concentration in the middle Proterozoic, as we thought back in the days, oh, well, it was all flat. Sure, nothing fluctuated, probably nothing was available for organisms to evolve, et cetera, et cetera. But again, it's a fiercely competitive sort of situation where we're constantly fighting. Was there oxygen? Yes, there was. No, there wasn't. If you look at the papers, they're quite entertaining, actually. So there's a lot of debate still that surrounds us, but it's, you know, a lot of focus on oxygen. I'm not sure why, because if you look at some papers, oxygen is not the most important element. Some primitive organisms or modern analogs of primitive organisms don't even require that much oxygen. So should we be focusing on oxygen as much? Sure, it's responsible for atmospheric oxygenation back in time when stromatolite communities slowly used light to produce, practice photosynthesis and produced oxygen. But, you know, organisms don't always necessarily require oxygen. Quite often they produce it. So perhaps we needed to look at it in a different context. And it's not, there's no guarantee that as soon as you increase the amount of oxygen in the atmosphere, immediately organisms will be like, great, there's oxygen, we're going to evolve. Oxygen can be very detrimental to species as well. So especially because of the formation of reactive oxygen species. So how did the organisms back in the day when they were first introduced to oxygen, how did they react and how did they find out defense mechanisms so then they could finally end up using oxygen to their advantage rather than disadvantage? And one of the problems of oxidation or reactive oxygen species is actually aging. And to give you a live example, this is me in 2017 and then 2023. As you can see, reactive oxygen species is not to be taken lightly. And just for fun, I took one yesterday and I was looking at my talk this morning and I was like, oh, maybe I should not have done that. So that was yesterday in my office. So no, reactive oxygen species is a thing, and organisms have had to develop strategies to not just a reactive oxygen species, but increase in oxygen also affected bioavailability of other elements that organisms use. So we needed to sort of think about, well, maybe too much oxygen is not very good for you. Temperature, I've been working with a thermal statistician who's looked at what kind of temperatures organisms survive in. And so how did temperature change in our oceans? How did that affect all the chemical reactions in our oceans? So that needed to be looked at as well. And so obviously we've had a general decline in the temperature, but that would have also had an impact on chemical reactions, etc. Mineral deposits through time, I'm obviously going to go through that in my talk tomorrow. But a lot of economic geologists also jump on board and say, yeah, very boring. See, there's nothing here. But like, oh, hang on, if you got to just look at it from a different angle, different plate tectonic settings, you see there are actually plenty of mineral deposits, sediment hosted, zinc lead in particular, some of the world class being here in our own backyard in the Northern Territory. So I've been working on some samples, and this is a paper that we published in Earth and Planetary Science Letters, where we start to see really good evidence of availability of nitrogen close to the hydrothermal venting sites, aka mineralization, as opposed to if you move away. And that's also demonstrated in some of the textures that we come across in our samples that are potentially pyritized microfossils as well. So there is a link between that mineralization and sort of diversification of life as well that we are working on. So here's are some of the textures that we come across. I just thought I'd show you some of them. Nutrient availability. Nutrient availability has also been looked at by several researchers, and some say it's all flat and boring, so no wonder nothing happened. And this is where our study or my study fits in. I'm part of a group, so I can't really just claim that that's all my study, but my PhD particularly focused on the distribution of trace elements, so the bioessential trace elements and how that changed during the boring billion. So we did have some previous studies, of course, and some trace elements were studied here, and they said, well, it's all flat and it was all modeled. But we thought, well, let's go and have a look at the essential key trace elements and see how they've varied through time. The other thing that I really wanted to focus when I was doing my PhD was, well, what did happen? Did nothing happen in terms of biological evolution? Or did something happen and we just don't see bigger animals? Is that the problem? So when you look at it and you think about the milestones in early evolution, the absolute milestones that underpins all biology on Earth today. Without those milestones, you and I wouldn't be sitting here. So the transition from a prokaryotic cell to a eukaryotic cell. If you speak to any biologist, they'll tell you how important that transition will have been. Of course, multicellularity and origins of sex. Combine these, eukaryotes have, evolved in a totally different route compared to prokaryotes because of these eukaryotic specific traits. And they've been extremely important to build any kind of eukaryotic evolutionary patterns. And when we started working on this, I wasn't necessarily involved with a biologist, but now that I finished my PhD and postdoc, we've had the time to write a manuscript together, which we just submitted like a few weeks ago. And it was a really good experience for me as a geologist to work with a biologist on this time span, where geologists keep calling it the boring billion, whereas when you look at it from a biologist's point of view, they can't understand, for the love of God, why we think it's boring. And particularly if you think about the evolution of the eukaryotes, the complex cell that you and I are made of, 34 trillion of which you and I are made of, evolved around that time. And that was very, very important. So biologists are tearing their hair off their heads trying to figure out, well, how it could have happened. Did two cells merge together in an endosymbiotic or a friendly fashion, or was one engulfed by the other? Was it predation or phagotrophy, if you want a fancy scientific word? How did it happen? And that's something biologists want to discuss about. But I think as a geologist, I want to see why it happened. What kind of conditions did Earth facilitate, or our oceans facilitate, for those interactions to occur, whether it's predation or whether it's endosymbiosis? And so another thing that the biologists we propose in the paper is that some of these evolutionary transitions that occur are intracellular and genetic. You may not have a representation of it in the rock record. So just because you can't see it in the rock record doesn't necessarily mean that it's not important. So we've gone through these little steps that are required to actually have a eukaryotic cell in the first place. And by all means, it can't be considered as boring at all. So there were lots of evolutionary breakthroughs that did occur during the boring billion. So just to give you an idea what endosymbiosis is just, I'll play the video while I explain. Will it work? No, it doesn't work sometimes. Doesn't matter. So it's the host is the paramecium, and those little bits in there are zoochlorella. And so one provides the safe space, the other is photosynthetic, so generates the food and energy for the host. So it's a friendly relationship. We see that today in our organisms all the time. The gut the microbes in our gut are practically the same thing here. Predation, yes, again, you can have examples of that. But when did these traits evolve is the question, and why did they evolve? Because these are the processes through which one cell acquired that complexity that you and I are made of. So when did these things occur? We are fairly sure of, which is, the Mesoproterozoic. And how did it occur? We can debate about it. But I think at this stage, what we're trying to do as geochemists is see why it occurred. Trace elements, really, really important. And so that's why we wanted to look at, well, let's see what happens using our proxy to see what happens how trace elements evolve through time, because they're really important for metabolic reactions as well. And I'm sure you understand this graph really, really well. Look, I'm not going to pretend, but I'm just going to demonstrate how complex these reactions are that sustains life on a regular basis. They run like clockwork. And so if you didn't have trace elements and the catalyst, these reactions would fall apart immediately and everything would just stop. So really, really important for us to understand how these elements and their availability varied through time. And did that have an impact on our organisms? What's my technique? I look at marine black shells through time to see if that can give us a measure of what trace elements might have been available through there. Why marine black shells? Because we are comparing anoxic portions of our oceans through time. And do we look at whole rock? Well, you could use conventional techniques, but we developed the pyrite technique, which is looking at early sedimentary pyrite, inorganic matter-rich marine black shells, through time. So it's not just randomly picking up any black shales and zapping the pyrite. So there's a whole body of, validation of technique, which I'm happy to address after the talk. But so far, we've been able to generate around 10,000 pyrite analyses. And that gives us reasonable confidence that we are starting to see some form of trend, which is subject to change. Absolutely, it is. So yes, although I have shown you an example of an outcrop along the beach somewhere in England, really, we want to use drill core samples because pyrite oxidizes very quickly. So these are the sort of pyrite textures that we've analyzed for the different trace elements using the laser ablation and generated the database. What do we find? So this was published in 2018. We did have a follow-up paper with additional data. But what we do see, first off, is that we're seeing the nutrient availability through time, and it's not all flat. That's the first thing that we noticed, that prior to what previous studies said, that must have not fluctuated a lot or something. No, that's not true. There was a huge sort of trough at the start of the boring billion, and then it recovered. That's all we can tell, or I'm reasonably confident in saying. And it's a matter of how you view this trend. So I'm not suggesting that that was detrimental or not. I'm just saying, compared to previous times, we obviously start seeing there's a massive decline in the trace element concentrations. And then finally, there's a recovery, which is not very different to other studies where they say, well, it was all very low and it didn't recover until much later. I'm just saying, yeah, sure, it was pretty low to begin with, but then it recovered. And I think a lot of the papers were claiming that this low period somehow delayed evolution, but I think I'm on the other camp. I think we needed this low period. I think without this low period, you're not getting a very balanced opinion from me, obviously, but here's what I think. I think this low period was absolutely essential, and we absolutely needed it to form the first complex cell, because this low period, or in other words, nutrient scarcity, prompted interactions such as predation or endosymbiosis. Otherwise, if two organisms are completely capable of living independently, why else would they come? So there was some sort of environmental pressure, and I think this nutrient low period might have contributed to some form of environmental stress, forcing organisms to come into interactions like that. There was the follow-up paper as well that we looked into, where we actually looked into what trace elements are usually taken up by organisms, and we think of life as this one thing, but they are incredibly smart, particularly microorganisms. If zinc's not available, they will be very happy with cadmium. If molybdenum is not available, they'd be very happy with tungsten. And in the process, they probably evolve or not, but there's a mix of things going on because they're used to substitution. I mean, knowing microbes, they've basically had command on Earth since basically the beginning of the Earth. So they know how to manipulate or substitute for trace elements if they're not available. So these low periods might have triggered some form of interactions. It's not that we don't know that. If you look at modern studies, they will tell you that sort of nutrient stress periods can sometimes increase genetic mutations in bacteria. So there are experiments that are being done. So I didn't just, dream these things. There are evidence supporting my hypothesis. And there are these amazing natural experiments as well, and observations in the natural world, where they've noticed that cooperation is more facilitated in sort of colonization of harsh environments. So you see more cooperation when environments are harsh, and this has been noticed in birds as well. So lots of different studies have pointed to the fact that sometimes stress can be really good for you and can lead to wonderful things. And I have absolutely no doubt that that might have happened millions of years ago as well. And the thing is that even though you think, well, we don't have much evidence of evolution of life, well, no, that's not true. As we go, just like the oxygen curve is constantly being revised, we're finding more information on evolution of life on Earth. There was a paper by Bengtsson et al. In 2017 that found some really important complex structures suggesting the first plants might have evolved in the boring billion. There are other papers that are also commenting, starting to find with new technology. I guess our ability to scan through the rock record gets better, and we're uncovering more and more new papers. The famous Rocks guy also happens to publish a really important paper also pointing towards the fact that we did have eukaryotic complexity. While we couldn't see that in the rock record earlier, we can see it now because we're looking at it differently. We're looking at different techniques. We're trying to understand it from a different viewpoint. So yes, it's not just like that nothing happened and then something happened with the Cambrian explosion. I sometimes think of the boring billion as, I don't know, a pregnant pause or something, where you basically, you know, the 9 months, sure, nothing's happening, and everyone's making a hoo-ha about the baby at the end, sure. But like, hey, you do need the 9 months, otherwise there would be no baby. So I think this is the pregnant pause before the baby. So you could call it the pregnant billion if you want. But that's how I view the boring billion. So you could then say, well, what about macroscopic life forms? If you had eukaryotes, because that's the new agenda these days. Well, you had eukaryotes. Why didn't we have animals? Why didn't we have macroscopic animals? Everything is microbial. So what? Everything is microbial. We are nothing compared to the microbial world. We are this one dot. This was in a museum in Harvard, which I thought was extremely exciting. This one dot represents animal and plant diversity, and this is what microbial diversity is like. And it's absolutely massive. We barely know anything about it. We're still trying to figure out. And if you think microbes aren't important, well, that's, you know, everything. There's more microbes on your face right now. There's more microbes in us right now than the number of cells in our body. I don't know why we have to view the microbial world as, oh, they're not sophisticated. Oh, they're very sophisticated. They basically run everything on the planet right now. They're small, but they're very, very, very significant. So there's a lot of microbial diversity that probably occurred evolution as well in the boring billion, which is completely going under our radar. And we think that, oh, nothing happened. There's no microscopic life forms. And it's important for us to understand how the microbial diversity has changed through time. I know we're worried about the climate and we're worried about us and our displacement, et cetera, but we also start to think about how the microbial diversity has changed through time and how it's changing, because it underpins the food chain in on our planet right now. So depending on how the lowest and the most diverse forms of the food chain behave, that will absolutely have an impact as we go up the food rank. So that is really, really important. Yes, sure, we haven't found any macroscopic organisms in the boring billion, but when was it? First of all, it was thought it was 540 million years ago. Then it was pushed back to around 800 million years based on molecular clocks, etc. Then you start finding vase shapes, microfossils, etc. So it's just a matter of time when we'll find more information and we'll be able to establish a link. So where I'm getting at with this is that there's no delay. In my opinion, there's no such thing as life was delayed or something. It's just the natural progression of things. We're just uncovering information at different rates. Everything's happened for the reason. You couldn't have macroscopic life if you hadn't established a fully functioning microbial system. Unless that's set up, you can't actually sustain macroscopic life anyway, because bulk of the oxygen that's being produced today comes from our oceans, is from the microorganisms. They don't need oxygen. They're producing oxygen for us. So until that was set up, I don't think we could have had macroscopic organisms anyway. So how much time have I got? Because I can't stop here.
I got some time.
Just maybe 5 more minutes. Is that all right?
Even 10.
Even 10? Okay. So where I was getting at is that we also looked at some other trends, and we started using AI because, I don't know, I guess we have to use AI these days. So we were looking at stromatolite trends through time, because instead of looking how this evolved through time, how that evolved through time, how about we like start comparing things and see where we get at. So I've always been fascinated by stromatolite sequences through time, the abundance of stromatolite sequences in the stratigraphy through time. So someone did it. Peters et al. 2017. They did a marvelous job of collecting all that data. And here's the graph. Here's how stromatolites have evolved through time. Now, the one that we love talking about is this one here, this little sharp drop. Oh, that's because animals evolved at that time. So it's not like stromatolites weren't there, but because animals evolved, they eroded all the stromatolites, that microbial mats. And so this is a preservation thing. So once you started having burrowing and grazing by animals, you stop preserving stromatolites. And so you start seeing this decline. But I was always interested in this decline here as a whole, not just this little sharp little thing down here. So we thought, well, stromatolite abundance through time, what encourages stromatolites to grow or not grow? What are some of the big picture trends that's available in the literature? And can we use machine learning to do, I don't know, some sort of comparison? So what we did was, I thought our data was good enough to get an estimation of how nutrients varied through time. We do understand how temperature varied through time. And if you look at microbial mats and what they require, et cetera, it's usually light and nutrients. There are other things like water clarity, radiation, et cetera. But I don't have the data for that, unfortunately. So this has to be taken with a pinch of salt and other things, of all things. So this is just a first try. This will obviously be canned, I'm sure, with new data or something. But it's a start to look at things more holistically. So how do we, so we did something called predictive modeling to see how, if we can see some sort of predictive capacity of nutrient variation through time and temperature, and see what best predicts the stromatolite abundance at any given time. So is stromatolite increase is a function of low nutrients, high nutrients, decreasing temperature, increasing temperatures, this sort of analysis. So yeah, I was involved with Ross Cochran, he's a statistician, and he did, we sat together and he was like, here's what it looks like. So what did we do? So we're looking at a decrease in stromatolites here, and I think it could be prokaryotic competition, because sometimes, you know, prokaryotes can compete for nutrients within amongst themselves as well. So you can see minor fluctuations, but it's more or less the same. So there's this whole body of work that's being done on prokaryotic, eukaryotic, symbiotic relationships, particularly microbial mats and eukaryotes, because of oxygen concentration. So that's where I think the oxygen link makes sense to me, that there's low oxygen, something requires oxygen, they haven't got it. So basically microbial mats are like a safe haven for the eukaryote. So there's this microbial eukaryotic, prokaryotic relationship that can be established during this time. And then you see this sharp decline. And again, this decline here in stromatolites, I attribute that to eukaryotic diversity, because we know that for a fact that we see lots of eukaryotic diversity in the rock record. So it's undeniable. And if you see, there's a significant rise in stromatolites here. And I think, again, it comes back to our oxygen in the atmosphere, where we want the earliest metazoans perhaps evolved around that time. If it did, I don't know if it did, but there's some reason why these went up. And I think it went up because it was in their favor. So what we're seeing here is differences in ecological interactions. We think that this rise here probably meant it was in the best interest of whatever was there. We don't know if it's metazoan or not, but it's in their best interest to have them live. So these microbial mats could provide as a safe haven. And perhaps that was one of the first instances in Earth's history where organisms have started to, you know, basically try and figure out what kind of ecological interaction works best in their favor. And then obviously you start this non-recoverable decline when animals absolutely diversify. So that's how you could interpret this trend just by looking at the data, but we modeled it using predictive modeling techniques, using trace elements in oceans and temperature. And we noticed that so different trace elements were important in different, we can't say whether it's the increase or decrease. You can comment on that because we know whether it increased or decreased. But all we know with these analyses are these variables were important. So temperature was always important in all time periods. Different elements were different in different time periods. But this one, between 1000 and 1300, this is when a lot of elements started to pop up in our predictive modeling, implying that these must have been important for stromatolite variation through time in some way or the other. And that's probably because that's when you started to see some more intense ecological interactions through time. So rather than focusing on this decline here at 800 million years, I think it's this decline that we need to focus on when it comes to Metazoan evolution. That's just a proposal. And sure enough, biologists or paleontologists are doing whatever they're doing amazingly, because they're discovering, or they think they have discovered, sponge fossils from close to 900. I wouldn't be surprised if someone finds something around the 1050 mark soon. But that's just, again, futuristic on my behalf. So the amount of information we are getting out of this time span is incredible. And the more work we do, the more information we get, the more our views are changing. And so I was just highlighting the more important bits of this article, really. But this is where I'll end and let you make your own informed decision on whether you think the billion year is full of boring things or full of brilliant things or balanced. Brilliant in my opinion, but I'll leave it up to you. Thank you.
Do you have any questions? You look at your question.
I have lots.
That is exactly what happened in the first.
So pick the best one and go.
Can you show us lots of good stuff in there? Can you show us the stromatolite curve again? I would like first to understand. It says proportion of what? Is this carbonate units?
Carbonate units, yes, but stromatolitic bearing carbonate units.
I would like to know which authors made that curve. How did they interpret it?
Yeah, so one of the limitations was that these are only stromatolites from the North Americas, I think. Just not the whole world. So that's a huge drawback. I think the software they used was called macrostat, and they literally went through, I think it goes through these different journals. It was quite thorough, I have to say. I know it's just North Americas, but it was very close to someone else who had who was it again? I think it was Nielsen 2013 paper. They also had a stromatolite abundance through time, but it was very sparse. And but that was more peppered across the world, whereas this one was just North America, and they basically looked at different papers, and they used the software to figure out who's published what and build a whole database from just that. So based on published literature only, yes.
But what did they say? I mean, the obvious things to assume is, for example, it's simply shelf area. The area of carbonate platforms in total, how deep is the water? That would be first control of stromatolite. Did they, for example, this excursion at point number 4, that's very, very smooth. Could that be a sea level change curve in North America, for example?
I'm not sure. They, from this graph, was supposed to be when they saw stromatolitic bearing carbonates increase or decrease in a stratigraphic sequence in mostly intracratonic basins. So passive margins maybe. So yeah, it's not a holistic picture for sure, but that's what we have right now. And why did the stromatolites increase or decrease? Yeah, so that's something that when we look at it, using predictors such as temperature and trace elements, I think sea level changes is something we could factor in. So that's another thing that could go into this model where we say, well, what were the sea level fluctuations like in the past? But wouldn't that already look after the proportion in the rock record anyway? I don't know. I mean, not that we know everything. This is not the holy grail, but it's a start. So we could look at things that way.
Thanks for the interesting talk, Matt. Your pyrite data is great availability. And any idea what's driving those?
Yes, yes, which is why I've had to delve into the whole plate tectonics side of things. And in this review paper that we did, we actually tried to explain just that very question, is that what's controlling that? So when I started the PhD, the idea was not to look at evolution of life or anything. It was oxygen in the atmosphere. So everything we saw in our data, we were tying it back to, well, that was because of increased concentration of oxygen in the atmosphere. But I think over the years, I've come to believe that it can't just be oxygen, because you could have plenty of oxygen in the atmosphere, but if you're not eroding the right rock type, or eroding for a start, if you don't have erosion, in other words, plate tectonics or mountain building activities, then you might have all the weathering, but it'll just be sat in one part. In order to move it, you need the erosion. So we definitely want to know what the geological regime was during the deposition of those black shales, the provenance. So what was being eroded? So you could have all the oxygen in the world, but if you're eroding a felsic rock or a continental crust of felsic composition, then you might not deliver the nutrient trace elements. So I think it's a combination of increased oxygen in the atmosphere, mountain building or high erosion rates, and source rocks. So it's a combination of all these things. But having said that, I'm currently involved with international ocean drilling projects, and I'm now looking at a lot of modern oceans and modern sediments, and how modern sediments are capturing modern ocean chemistry. And I'm starting to think, my God, there's so many things. Even if you deliver the trace elements in the oceans, where you are depositing them is really important. So we compared shallow versus deep water environments. They're very different. We did that in the past. I think the shallows are consistently or distinctly very different to the ones that are more deep. As you go deeper in the basin, you look at different drill holes, they're more or less constant, but the shallower drill holes, they always show very different traces. So we have to do all these things before you start joining the dots. But yeah, it's a combination of what happens on land and then what happens at the sediment water interface as well.
George?
Great talk. I agree. I don't think that the period between 1.8 and 1.3 was boring at all. In fact, if you look at Northern Australia, there's very good evidence, I think, from organic geochemistry, but also from mafic rocks, that we had 3 cycles of subduction, advance, and retreat, ultimately leading to an ocean closing. So during periods of subduction advance, we get basin inversion, uplift and erosion. And again, as you were arguing, that's when you get some of your nutrients spreading into the oceans. But during times of retreat, things essentially collapse, you back into basin formation, and different tectonic setting, and probably a different set of nutrients finding its way into the ocean. So I just wanted to say, I don't think it was boring at all, quite the opposite.
Thank you.
I think in the balance paper, there's a good point, is the transition from bottom up to top down.
Yeah.
From kimberlite to commodite. I think that's a very smart way of thinking. Like in transition, balance,
mantle convection. Yeah, I think the
point of seeing the
yeah, I absolutely agree. It's like I said, I think the more we look at it, yeah, you definitely don't want to see those photos. Yeah, so I agree. I think it's I mean, knowing Earth, seriously, the more I think we're very naive in sometimes when we propose things, I guess. And I've proposed a lot of things in my presentation as well, but I'm very happy to be proven wrong. And also the other thing is that we have this idea that everything we look at is a global signature, but different things happen at different parts of the oceans at different times. So how relevant it is that we always want to extrapolate our data from being local to global, I'm not quite sure. So yeah, while this is, we always like to have our x-axis as age, and everything we show is perhaps representing a global signature, but maybe it was quite heterogeneous and then evolution occurred at different parts of the earth at different times. We could definitely consider that possibility.
Time for one last question. You okay? Short one?
I would have another difficult one.
Sure.
I'll answer first. How could, start with the Redfield ratio, 1/6 to 1, the ratio of carbon, nitrogen, phosphorus, in sediments and living things. And it would be tempting to say, hey, this is the ratio in which Earth supplies the elements to life, and to our life incorporates in that ratio. But in fact, that ratio exists because that's how nature picks those elements and puts them into their own biomass because it works well.
Yeah.
So in that sense, if something is not a biolimiting element like phosphorus usually in the iron or other things, how could trace elements have any impact on evolution of life at all? And could their concentrations through time rather be a consequence rather than a cause? That means reflecting which elements they want rather than constraining what they can do.
Absolutely. So Redfield ratio, so organisms can deviate from that. And it only happens, so this I can give you the reference. And in fact, there was a Nature paper on 80 years of the Redfield ratio. There was a whole thing on that. Organisms can deviate today in modern oceans, deviate from the Redfield ratio only under low nutrient conditions. So that's a known fact. So that can definitely happen. With regards to trace elements, sure, we need the macro elements. There's no, it's an undeniable fact. But by definition, macro elements are present. I don't know what's the definition. I think it's more than 1 milligram per dry matter or whatever. But their concentrations are usually higher. But even if you had higher concentration of macro elements, so this has now been found in the Southern Ocean, where you have really high phosphorus, but there's nutrient limitation because of iron. So it's not as previously thought that you need nitrogen and phosphorus. Yes, absolutely. But if you're missing some of the trace elements, then that's tied in with how the organisms respond to the availability of macronutrients as well. So sure, trace elements alone are not going to answer the whole question for evolution. But what I'm saying is that they're a big part of it. I am guessing that phosphorus and nitrogen will have been, because if you look at Eva Stoican's papers on nitrogen isotopes, or Reinhardt et al. For phosphorus, it's all there. It's not like it is very low as well. They just say it's flat. So perhaps they were enough. But I'm not saying that you can do without macronutrients. All you need is trace elements. So absolutely not suggesting that. It's an important part. Yeah.
Let's take again. Okay, a reminder, there's a sign-up sheet outside for dinner, 6 p.m. lemongrass. I'd love to have you there.